remarkably. So up to about 10% of the patients who undergo renal transplant will eventually develop PVAN due to BKV reactivation. And up to 90% of the patients who develop PVAN will eventually lose their graft due to BKV reactivation. So this virus is a huge problem in those patients. But unfortunately, there's currently no specific, efficient anti-BKV treatment available for the patients. And usually the physicians will choose to reduce the amount of immunosuppression to control for virus reactivation. But that might leave the patients at risk of graft rejection. So one of the goals in our lab is to gain a full understanding of the interplay between the virus and the host cells. So hopefully it will allow us to identify potential better anti-BKV treatment options. So here now let's look at, on the molecular level, what does the viral genome look like? As you can see here from this schematic view, the genome is small and can be divided into three major parts. So on the left is a region called early coding region. It encodes three proteins that we know so far. One is the large T antigen, the second is the small T antigen, and the final is a recently discovered Trump T antigen. So just keep in mind the basic function of these proteins is to set up an ideal host environment for the virus to replicate. Okay. On the right is a region called late coding region. It encodes the viral structural proteins, what the viruses are made of. It includes the major capsid protein VP1 and the two minor capsid proteins VP2 and VP3. So the virion is made of 72 VP1 pentamers, and each VP1 pentamer associates with a single copy of either the VP2 or VP3. And there's also a fourth protein called the actin protein present in this region. The exact function of this protein is not entirely known, but it has been involved in the egress process of the virus from the host cells. And in the middle is a third region, which I'll be referring to as non-protein control region, or NCCR. So this is the region contains the regulatory elements for virus replication, such as origin of DNA replication and the promoters for both the early and late transcription. So how does the virus infection occur at the cellular level? So it begins with the virus binding to the cellular receptors. We know now it's granulocytes present on the cell surface. And after the binding, it then gets internalized into the host cells. And it has to go through a series of trafficking events. And the goal of this trafficking is to deliver its DNA genome into the host nucleus. And this is where everything happens. So the first thing happens is the early transcription, which leads to the expression of early proteins. As I mentioned, the large T antigen, which is the major regulatory protein present in virus life cycle, it does so many things. So one of the things it does, it translocate back into the nucleus. It binds to the origin of DNA replication that is present in the NCCR region, initiate viral genome replication. And the second thing it can do, it can also bind to the late promoter, initiate late transcription. And the virus assembly begins with the capsid proteins or the late proteins, translocating back into the nucleus, encapsulates the newly replicated viral DNA, and then forms the virions. And the final step of the virus life cycle, which is the egress of the viral particles out of the host cell, at this point is not clear. That's why it's not depicted here. So in order for us to study BKV infection in vitro, so currently there's no good animal model for this. So most of our studies that we've done is using a tissue culture system that we previously established using primary human renal proximal tibial epithelial cells, which I'll be referring to as RPT cells during the rest of my talk. So we have shown that a BKV strain that we use in the lab is able to establish a lytic infection in this cell type. A lytic infection means it can go through a complete its whole life cycle in this cell type that we use in vitro. So what we found is that if we infect those cells with purified virus, we can detect early gene expression at about 24 hours post-infection. And then by 48 hours post-infection, we can start to detect viral genome replication, late gene expression, and also progeny production. And in addition to these molecular events, we can also observe morphological changes that are present in these cells, including the cells rounding up and the presence of these large nuclear inclusion bodies, which are very similar to those seen in PV and biopsy samples. So these experiments established the cell culture system as a very useful in vitro model for us to study lytic infection between the host and the virus. And most of the experiments that I'll be describing today were done in this cell type. So when I was a kid, when I think about virus infection, at first I always thought it was a one-way street. The virus gets into the cells. Somehow it starts the infection, eventually kills the cells. But the more I study the virus-host interactions, the more I realize it is really an ongoing battle between the virus and the host in terms of interactions. And in the case of BK virus, you can imagine there could be two potential battlefields. One is in the cytoplasm. I already told you that the virus has to traffic all the way from the plasma membrane to the nucleus to establish infection. And along this journey, the host may sense the presence of the virus and potentially mount an innate immune response to counteract this virus. And for BK, I think the major battleground is happening in the nucleus because, as I told you, this is a place that everything happens, including the viral gene expression and the genome replication and progeny production. And how does the host achieve to prevent all these things from happening is also one of our research areas. So today, the stories that I'll be telling you is mostly focused on interplay between the virus and the host nuclear components. And if you look at the components that are present in the host nucleus, you can imagine there can be potentially two types of factors that might affect the outcome of infection. The first, I like to call them repressive factors, meaning they might have antiviral functions. So in order for the infection to proceed, the virus has to either disturb them, inhibit them, to establish this infection. And in the second category, I like to call them facilitating factors. They might be normal cellular processes or pathways, but somehow because they're beneficial for the virus infection, the virus might be able to hijack the normal cellular processes and use them for its own benefit. So one simple example, I already told you, the virus genome is very small, it's only 5 kb. It doesn't encode its own DNA synthetic machinery. So in order for the infection to occur, it has to drive the cells into S phase. This is where the host DNA synthetic machinery is activated, so that it can be used by the virus for genome amplification. So today I'm going to tell you one story about one of the repressive factors that we were looking at, and also another story about the facilitating factor. That will be the second part of my talk. So before I go into the actual data, I'd like to introduce you several assays that we use to monitor the infection process of BK in RPT cells. So to monitor for early gene expression, we use either Western blot or immunofluorescence microscopy using a specific antibody that recognizes large T antigen to look for the expression level as well as the localization of this protein in the nucleus. And then we also have a quantitative real-time PCR assay to quantify the amount of viral genome that are present in a given cell. Or we have established a fluorescent in situ hybridization, also known as FISH, to look for the localization of the newly synthesized viral DNA that are present in the nucleus. And finally, we have an infectious unit assay or IU assay to quantify the amount of infectious particles that are present in a given lysate. So pretty much if you have a lysate, you can use them to infect naive RPT cells and quantify how many T antigen positive cells that are present in those cells, and then give you an idea of how many infectious particles are present in a given lysate. Okay. So the repressive factor that I'm going to tell you today is called the promyelocytic leukemia nuclear bodies, also known as PML nuclear bodies, nuclear.10 or PML oncogenic domains. So what are those nuclear bodies? So they're basically subnuclear structures that has a very heterogeneous protein component. And the major protein component of this subnuclear structure is called the PML protein. And this protein has been involved in the development of promyelocytic leukemia, and that's where the name came from. If you stain the cells using a specific antibody that recognizes this PML protein, then you can visualize the presence of PML nuclear bodies that are present, which is shown here on the right. So they appear as small, bright foci that are present in the nucleus. So there's a really long list of proteins that have been found either associated with or present in the PML nuclear bodies. Some of them are considered constitutively present, including PML protein itself and the two other proteins, FLAX and SP100, that have been implicated in regulation for transcription. The subproteins, they're only found in those structures under certain conditions, for example, different overexpression systems or stress conditions, and they might manipulate the function of PML nuclear bodies. And the reason that we became interested in looking at the relationship between this nuclear structure and the virus is because these PML nuclear bodies as implicated in both the intrinsic and also the interviral-inducible antiviral functions for a number of DNA viruses, including herpes viruses and adenoviruses. And in order to inactivate the antiviral functions of these nuclear structures, different viruses, they come up with different ways to do that. For example, adenovirus is known to reorganize the PML nuclear structures from punctate into track-like structures. And for herpes, things are even more dramatic. There is a protein called ICP0 that is encoded by HSV, is able to degrade the PML protein and thereby disrupt the functions of PML nuclear bodies. So when we first started this project, we just want to ask a simple question. What are the effects of PML infection on PML nuclear bodies in RPTE cells in our relevant cell type? So here you're looking at a simple experiment where we just either mock infected or infected cells with BK purified virus at a high MOI, and then we double immunostain the cells for either the T antigen, which is shown in red here to allow us to identify positive BK positive cells, or PML, which is shown in red here, in green here, to allow us to visualize the presence of PML nuclear bodies. As you can see that in the BKB positive cells, there's a dramatic change in the morphology of PML nuclear bodies. They seem to be bigger in size and fewer in numbers compared to either the uninfected or the mock-infected cells. So we have quantified our results here on this slide. So on the left, we're looking at the average number of PML nuclear bodies per cell with the mock-infected normalized to 100%. So you can see that compared to the mock, there's about 60% drop in the average number of PML bodies per cell when the cells are infected with BK. And on the right, we're looking at the size distribution of those nuclear structures. So showing you is the mock, you can see that most of the PML bodies are pretty small in size, the diameter of which is about less than 1 micron in size. But however, when the cells are infected with the virus, we start to detect really big PML nuclear bodies. Some of them, the diameter is over 1.5 micron. So the question that came to us is what caused those PML bodies to become so big? And then what are the viral components that contribute to this reorganization process? So to get a first clue, what we first use is to look at the relative localization between different viral components and the PML bodies and see if that can give us some clue as to what contributes to the reorganization process of these structures. So here you're looking at a time course of infection where we fix the cells over a 3-day time course. And again, we stain them either with T antigen in red or PML in green here. So you can really see the progression of the infection because the nucleus shown here, they're getting bigger. So these are all pictures taken at the same magnification. So if we look at the localization of the T antigen as relative to the PML, we can see that during early infection, this is when we first start to detect large T expression. They appear as small punctate foci in the nucleus, and some of them seem to be co-localizing with the PML nuclear foci. But starting from 2 days, the T antigen signals seem to get really diffused throughout the nucleus, and we start to detect a lot of areas that seem to be devoid of T antigen staining. We still don't know at this point what these areas represent. We think that might represent the areas where viral assembly occurs, but we still need to do more research to figure that out. But if you look at the localization of PML bodies, so first we need to notice that they're definitely getting bigger and fewer in numbers, and some of them at 3 days post-infection, they become so big, they almost look like ring-like structures in the cell. And the localization of PML bodies, the T antigen is not random at this point either. They seem to be localized to the periphery of these T antigen excluded areas. So this is what it looks like in terms of viral protein relative to PML nuclear bodies. So next, we also looked at the localization between the viral DNA and PML nuclear bodies. So here, I'm using fluorescent in situ hybridization to visualize the viral DNA in the nucleus. So I'd like to point out, because of the sensitivity of this assay, we're only able to detect viral DNA that are present either at 2 days or 3 days post-infection. So presumably representing unit-replicated viral DNA. We're not able to detect input DNA using this method. But what we can see is that in some of the early infected samples, because the nucleus is still pretty small at this point, we found some of the bright foci of viral DNA. And then the signals became more diffused by some of the late infected 2 days post-infection samples. And then almost reminiscent of the T antigen signals. You can see some of the areas are excluded. And then by 3 days post-infection, we can really detect some of the really big foci of viral DNA present in the nucleus. And if we look at the relation between the viral DNA and the PML bodies, what we found is they don't seem to necessarily co-localize, but they seem to be adjacent to each other in some of the late infected cells. So as I told you, we can see the co-localization between the T antigen and the PML nuclear bodies during early infection, and also the adjacentness between the viral DNA and the PML bodies during late infection. So what is the cause for the reorganization of this PML nuclear bodies? So whether is it due to T antigen expression or is it due to viral DNA replication? So in order to address the first possibility, which is whether T antigen alone is sufficient to drive the reorganization of this process, we use an antivirus expressing large T antigen, transduce that into the cells. Basically, it's a system to allow you to express T antigen alone without any other viral components in those cells, and look at the effects of the expression of T antigen on PML nuclear bodies in those cells. What we found is if we have T antigen present alone, compared to either the mock transduced or the cells that are transduced with anti-vector control, if we just look at the PML nuclear body morphology, there's really not that much difference between the T antigen positive and the T antigen negative cells. So this data suggests to us that T antigen alone is not sufficient for driving the PML bodies to become bigger. So now what about DNA? So in order to address the second possibility, which is the viral DNA replication somehow is coupled to the reorganization of PML nuclear bodies, what we did is we used a drug called Ara-C to block viral replication and assess the effect of the block of viral replication on the morphology of PML bodies. So first you can see if we just had mock infected cells, whether there's Ara-C present or not, there's really not that much difference in terms of the average number of PML bodies per cell or the size distribution of PML bodies. And I also have to point out that we have to adjust the MOI, basically the amount of virus that we use to do these experiments, because we want to make sure that the only variable that are present in the two samples are viral DNA replication. We want to make sure that all the other protein levels are similar, so that we have to use a high MOI when the viral DNA replication is inhibited. But what we can see is whenever there's active viral DNA replication, we can see a drop in the number and an increase in size. And whenever we have viral DNA replication inhibited, we can no longer detect those differences. They seem to be fairly similar to either the mock or the mock treated with Ara-C. So these data clearly suggest to us that the morphology change of PML bodies is due to viral replication, DNA replication, and not due to gene expression. So you may want to ask, okay, why does the virus bother to reorganize those structures? What is the point during its life cycle? So we hypothesize that because it's considered an antiviral structure, we think that the virus uses strategy to inactivate its antiviral functions during its life cycle. So how can we assay that? So to address this question, we turn to a surrogate, which is herpes simplex virus, to allow us to probe the antiviral functions of PML nuclear bodies, because there's really no direct assay to examine for that. So as I already told you, for the wild-type HSV, includes a protein called ICP0 is able to degrade PML protein and thereby inhibiting the PML nuclear body antiviral functions and therefore allow the virus to grow. Okay, so the wild-type can grow happily in the cells. However, if you have a mutant HSV, which doesn't express ICP0, so the virus can no longer disrupt this structure, and therefore it has a growth effect in the cells. So if our hypothesis is correct, that we think BKV infection reorganizes the PML bodies to activate its function, that it should be able to allow us to grow this ICP0 non-mutant virus in the cells, because the PML bodies are already disrupted. So no longer needs ICP0 to be present. And this is an experiment we did. So what we did is we first infected RPT cells with BK virus for 3 days to allow for the reorganization of PML bodies to occur. And then we came in, super-infected the cells with either the wild type or the ICP0 non-mutant of HSV. And then we harvest protein lysates, either probing them for ICP4 or GB, which is an early or late marker for HSV growth. So as you can see, whenever we're infecting with wild-type HSV, whether BK is absent or present, there's really no difference in terms of ICP4 or GB expression. expression. However, if we have BK present, and then we superinfected them with the ICP0 non-mutant virus, so without the virus BK being present, there seemed to be a defect compared to the wild type. And whenever we had BK present, there seemed to be a complementary effect. So this is consistent with our model, which is BKV infections disrupts the PML nuclear bodies and therefore allow the ICP0 non-mutant of HSV to grow better. So let me just summarize what I've told you so far. So we believe what's happening is that during early infection, somehow the viral genome is targeted to those nuclear bodies. And at this point, the PML nuclear bodies, they're still small, and there are quite a few of them in the cell that appears normal. And during late infection, as the viral DNA replication is happening, and then somehow it can lead to the reorganization of those PML nuclear bodies. And that is correlated with the disruption of the antiviral functions of PML nuclear bodies in the cells. So this is the first part of the talk. I'd like to pause for a minute. Any questions before I go into the second part of my talk? Yes? Oh, sorry. Okay, probably. I'll try to speak up a little bit more. Yeah. Any other questions? No? So the second story I'm going to tell you is one of the facilitating factors that we looked at during the virus life cycle. As I mentioned, we think the virus would either need to use them or promote them in order for the infection to proceed. So the response that we looked at is the cellular DNA damage response, also known as DDI. So this is a response that it's basically a signaling cascade pathway that is initiated by the cells when the cells encounter different types of DNA damage. And then the function of the signaling pathway is to allow the cells to lead to either cell cycle arrest, so that the cells have more time to repair those damage before mitosis occurs. Or if the damage is too severe, it can lead to apoptosis to get rid of those severe damage that are present so they don't get passed on into larger cells. And there are two key kinases that are involved in this response. One is called ATN, mainly responds to a very severe type of DNA damage called double-strand breaks, usually caused by ionizing radiation. And there's a second kinase called ATR. It responds to different types of DNA damage, mainly either UV treatment or replication stress. And again, this response has been involved in a number of DNA virus infections. And depending on the virus that you're looking at, the virus has either to inactivate or hijack this response for its own replication. And one of the reasons that we became interested is because a previous microarray study conducted by our lab has shown that during virus infection, several members of this DNA damage repair pathways, they have been shown to be upregulated in BKV infected cells. So that's where we initially triggered our interest to look at the relationship between the virus and the DDR response. So the first branch of the DDR response we looked at is the ATN branch. We're wondering whether the virus infection activates this damage response and what is the role of this response in virus life cycle. So as I told you earlier, ATN mainly responds to the double-strand breaks. And then once it's activated, it can turn from its inactive dimer to active monomer through autophosphorylation. And it can lead to the downstream activation of several signaling proteins and repair proteins, including H2AX, which is a histone variant, and it helps recruit DNA repair proteins to the site of damage and allows the cells to repair those DNA damage. And it also activates a protein through phosphorylation called CHEK2, which is a protein involved in cell cycle arrest. So here, basically, you're looking at a series of Western blots over a 3-day time course of infection, basically probing for the markers that are indicative of ATM activation, such as phospho-ATM or gamma-H2AX, and phospho-CHEK2. So what you can see is that as infection progresses, which judged by the T antigen expression here, we really detect a really strong activation of this response, including a strong activation of phospho-ATM here, and also an increase in both the gamma-H2AX and also the phospho-CHEK2 here. So this piece of data suggests to us that the BKV infection is able to activate at least the ATM branch of the DDR response. And as I told you, this response often leads to cell cycle arrest to allow the cells have more time to repair the damage. So the next thing we looked at is whether infection of BK leads to the cell cycle arrest in RPT cells. So here you're looking at the cell cycle profile and over a 6-day time course, either mock or infected. So in the mock infected cells, there's really not much going on over the 6-day time period. You can see that most of the peaks is around G1 and there's a small peak of G2. And there's really not that many cells going through S phase. So with the BKV infected cells, starting from 2 days post-infection, we can really start to detect the increase in the cell population that we call is larger than G2 population. It's more obvious by 4 days post-infection. So this is consistent, again, with the previous piece of data that the ATM branch is activated and then leads to the polyploidy of the cells that are infected with BK. So then you may wonder, what is the point of this activation? So to address that, we use siRNA to knock down ATM expression in RPT cells and assess the effect of this knockdown on viral infection, including both early gene expression, DNA replication, as well as progeny production. So you can see that the knockdown is fairly effective in our system, either judged by the ATM block or phospho ATM block. We can achieve up to 90% knockdown in the cells. And when we use these knockdown cells and then come on top of that with infection, what we found is that the absence of ATM didn't really affect the early gene expression. You can see that the tenogen level is fairly similar between either the no SI control or the non-targeting siRNA control over a 3-day time course. However, we then start to detect defects in viral DNA replication at 2 days post-infection. You can see that in the ATM knockdown cells, there's about 50% decrease in the viral DNA replication. And correspondingly, there's about 50% decrease in the progeny production. So this suggests to us that the activation, or at least the presence of ATM, is important for an optimal level of viral DNA replication. Because we saw the activation of this response, we're wondering if ATM is the main kinase responsible for this response. So here you're looking again at ATM knockdown cells, then we probe them for the markers that are previously shown to be indicative of ATM activation, including phospho-CHK2 and gamma H2AX. So you can see compared to the mock control, there's an increase in infected cells with both phospho-CHK2 and also gamma H2AX. And when we knock down ATM, at least for CHK2, there seems to be a decrease, suggesting that the virus activation activates ATM, then activates CHK2. At least this pathway is contributed to ATM. However, when we look at the gamma H2AX, really we didn't see a decrease when we knock down ATM. There seems to be, if anything, more activation of this important regulatory protein in the cells that are knocked down of ATM. So this suggests to us, so maybe this pathway is not what contributes to the activation of H2AX in BKV infected cells. So maybe there's another kinase that's responsible for the activation. So what can it be? And the obvious candidate is ATR. As I mentioned to you earlier, this ATR responds to different types of DNA damage, namely either the UV treatment or the replication stress. So when we looked at whether BKV also induces the ATR branch of DDR, the answer is yes. Because when we probe for the markers that are indicative for ATR activation, which is the phosphorylated form of CHK1, is another protein involved in cell cycle control. And here we see the induction of either the two markers that we used to look for ATR activation. And the next thing we did is to look for if we knock out ATM or ATR, or both, does that abolish BKV-induced DNA damage activation? So here you're looking at either the mock or infected cells with either control, or cells that are knocked down with ATM, based on this plot, or cells that are knocked down for ATR, or both. So as I showed you previously, whenever we knock down ATM, there seems to be a decrease in phospho-CHK2 level. And now we can knock down ATR, and what we found is that the CHK1 activation is gone. So suggesting that the BKV activates ATR, which then activates CHK1. However, when we look for gamma H2AX, surprisingly, with either the single knockdown or the double knockdown, we can still see a huge activation of gamma H2AX in BKV infected samples. So to us, right now, it's still a mystery of how this protein is getting activated. So we're thinking it might be something to do with PP2A, which is a phosphatase that is also targeted by the virus. But that still needs to be more research done on this one. So the next one we want to look for is what is the effect of ATR knockdown or the double knockdown on the viral infection. So here you're looking at, again, Western blots with double form of T antigen expression in either single knockdown cells or double knockdown cells over either 2 days post-infection or 3 days post-infection. What we found is that, again, with ATR knockdown, nothing seems to be happening with at least large T expression. But with the ATR knockdown, or when both were knocked down, what we found is that there is a top band that we usually see when we run T antigen Western blots, which we denote to them as T antigen star. That seems to get reduced whenever ATR is knocked down. And we think this probably represents a modified form of T antigen, and we're still in the middle of trying to figure out what this modified form is. But at least suggests that ATR may be responsible for some of the modification status of large T antigen. And what's also interesting to us is at 2 days post-infection, we can see that the presence of a third T antigen band showed up at 2 days post-infection. Again, we don't know what it is yet, but definitely leads to the direction that ATR is affecting the modification of T antigen. So then what about DNA replication or progeny production? What are the effects of ATR knockdown on those aspects of viral infection? What we found is that with the single ATR knockdown, it seems to have a similar effect as the ATM knockdown. There seems to be about 50% decrease in both the viral DNA replication and progeny production. And however, when we knock both down, what we found is that there is additive effect of these two kinases. Both seem to be contributing to an optimal replication stage for BK. So then we want to get into more mechanism of how these two kinases contribute to the virus infection. So the first thing we try to look at is whether either the knockdown of ATM or knockdown of ATR affects the distribution of the T antigen inside the cells. So that's the initial fact. So when we start doing this, we saw some very interesting results. So just to walk you through this. So if you just have regular mock infected cells, no virus present, you either knock down with ATN or knock down with ATR, nothing happens. The nucleus looks normal and there's no T antigen. However, if you have infection on top of the knockdown, we see some really interesting phenotypes. So if you have cells that are infected but not knocked down with anything, you can see, so here shown blue is the DAPI staining to visualize the nucleus. You can see most of them, they look normal, oval-shaped nucleus. However, if we knock down of ATM in the presence of BK, what we found is that in addition to those big oval-shaped nucleus, I don't know if you can see this, it's a little blurry here, is that there is some smaller fragments of DAPI staining present. We call them fragmented nuclei present in these cells. And the phenotype is more severe when we knock down for ATR. You can really see some of the really small nucleus present in the ATR knockdown cells. And what about T antigen? We also saw some interesting phenotype. In the cells that present some small nucleus in the ATM knockdown cells with the infection, you can see that the T antigen seems to be diffused throughout the whole cytoplasm. And then this diffuse pattern is also observed with the ATR knockdown, but with the ATR knockdown, we also can observe some of the really fragmented T antigen staining inside those cells. So it really messes up the T antigen expression in those cells. And here is the quantitation for the previous slide. So you can see with the mock infected cells, nothing much is going on with the knockdown. However, if we have infection in combination with the knockdown, we can start seeing a slight increase of fragmented nuclei in the ATM knockdown, but a dramatic increase whenever we knock down ATR. And also we start to detect very bizarre T antigen phenotype, which is increase in both the diffuse in the ATM or ATR knockdown cells or the fragmented phenotype with the ATR knockdowns. So what does this mean? The first thing that came to our mind is that because previously people had described the phenotype of these fragmented nuclei as micronuclei, that oftentimes are observed when the cells are undergoing severe DNA stress or DNA damage. So in order to answer the question whether what we see is a true reflection of the damage that is present in the cells, what we did is we used metaphysisgram to directly visualize the chromosomes that are present in those cells and see whether the appearance of the micronuclei corresponds to the appearance of damage that are present on the chromosome. So before I go into the quantitation, I decided to show you several phenotypes that we see in this type of experiment. So normally you can see the presence of nicely spread out chromosomes in normal cells. However, if you have cells that have a mild level of DNA damage present, you can start to see like breaks here in different parts of the chromosomes. And what's more severe is that if the damage is too complete, what you see is you can't really even see the presence of intact chromosome. What you see is what we call a shattered phenotype. Everything's completely shattered when you do those metaphase spread experiments. And this is actually what we saw when we have a combination of BKV infection as well as the ATM or ATR knockdown. So again, if you just have the knockdown without the virus, nothing happens. Everything seems normal, okay? If you have the virus but no knockdown, again, things are mostly normal. But you have the virus either with the ATM knockdown or the ATR knockdown or both. What we saw is that up to 30 to 50% of the chromosome that we scored, they appear as this shattered metaphase phenotype. And among the cells that are not shattered, we can also see a significant increase in the average breaks per cell that are present whenever the cells are either knocked out by ATM or ATR or both in conjunction with the virus infection. So this is what it really tells us is that there are two parts of the DNA damage response that plays during the BKV life cycle. So the first part we think what's happening is the virus infection activates the ATM and ATR response, which leads to the activation of both phospho-CHK2, phospho-CHK1, and ATR may also contribute to the modified form of T antigen. And that will create an optimal condition for both the viral DNA replication and also the viral progeny production. But the second part of this story is that we think the virus is able to induce severe damage on the host chromosomes. But normally, because you have ATM or ATR present, these damage are quickly repaired. However, when we knock down those ATM or ATR kinase, then that's when you see the severe damage that are caused by this virus. So just a summary of what I have told you in terms of the general picture of my research. We think there's an ongoing battle between the virus and the host cells. So I've shown you that BKV is able to interact with multiple cellular components during the infection. And also it can either hijack whatever is beneficial to its replication or perturb whatever there's antiviral functions to allow for the infection to proceed without any stop. So shown on the right is a cell that is infected with BK, and the red is positive for viral DNA, and the green again standing for PML nuclear bodies. And we all think that this cell is screaming for help that's infected by BK. In terms of the long-term future directions of these projects, I'd like to propose two aims for my future laboratory. The first aim is as a logic follow-up of what I've done in Mike's lab, which is basically determining the functional significance of both the PML nuclear bodies and the DDR response for BKV infection. So I'd like to figure out, again, what are the real viral components that drive either the reorganization of the PML nuclear bodies or the activation. What is really activating this response? Is it viral gene expression, or is it aberrant viral DNA structures that are present during the infectious cycle that leads to the activation of these responses? And we'd also like to further characterize the functions of other PML nuclear body components and DDR components. As I told you, PML nuclear bodies is very heterogeneous, and it has several constitutively present proteins that might be involved in transcription regulation. So we'd also like to dissect the functions of these proteins in the life cycle of BK. And finally, we think there's a potential link between the two processes. Because several DDR proteins have previously been reported to be present in the nuclear bodies under certain conditions. So wondering, is it because the virus can somehow make the link between the PML nuclear bodies and damage response, and the PML nuclear bodies as a surface to recruit those DNA damage response proteins to maximize its function during the virus life cycle. And the second aim I'd like to propose is to use powerful quantitative proteomics technique to really look at global changes that can be caused in those cells. As I've shown you, we looked at two structures so far, and both of them seem to give us very interesting results. So what about the rest of the nuclear structures or cellular components? So we just submitted a grant collaborating with Dr. Phil Andrews in the Department of Biological Chemistry, who is a leading expert in quantitative proteomics. So proposing to look at both the nuclear proteome and also the phosphoproteome in comparing the mock or the BKV infected cells, as well as to identify what exactly is interacting with this master regulator T antigen during different stages of virus life cycle. So with that, I'd like to give acknowledgments. First, I'd like to thank my mentor, Dr. Martin Howard, for his continuous support. And also, Nicole, the student who worked with me on the DDR project, contributed a lot to those experiments. And I'd also like to thank 3 undergraduate students that have worked with me throughout the years. Julia, Monica, and Justin. Julia is at UT Toledo going to med school. And Monica is a student at Southern Madison. And Justin is going to pharmacy school soon. And also I'd like to thank our collaborators. Without them, our research wouldn't be possible. So I'd like to thank Billy Tsai for collaborating with us on some of the tracking projects that I don't have time to talk to you about today. Tom Glover for helping us with the metaphase spread experiments. And also Dr. Thomas Staminger for providing us with a lot of reagents with the PML nuclear body project. Near DeLuca for providing us with the HSV reagent, and also at UVM, the microscopy core, and the Center for Life Cell Imaging for a lot of the microscopy experiments that I show you today. And finally, our financial support in NIH and also AHA postdoc fellowship to look at BKV infection. And with that, I'd like to take any questions.